So, good afternoon, all of you. In today's class, we are going to discuss about the locomotion and the movement chapter. So, many concepts we have to discuss in this class. So, we can start. So, you know that you might be very familiar with the movements. We will make the different types of movements, rotationary movements. Sometimes inside our body, there are so many movements. Similar to us, the other organisms also will make the movements. There is no doubt. So simply if you move this hand or the legs, you can just call it as a movement. But what is the locomotion? What is the difference between the locomotion and the movement? We can just see that. So when a movement will become the locomotion? So that we can see first. So all of you please look into that. What is the locomotion and what is the movement? We have to first understand that. So just imagine your movements are making some displacement of your body. Then you can call it as a locomotion. So let me repeat. Your movements are making the displacement of your body. Then you can call it as a locomotion. So for example, I'm just walking here. It's a kind of locomotion because my previous position was this. From that, I got a displacement. So it's a locomotion. If I jump, it's a locomotion. So if I run, it's a locomotion because all those cases, there is a displacement of the body. So I would like to write the definition for the locomotion. If the movements bring the displacement of the body, then we can call it as a locomotion. Like the jumping, walking, running, etc. But the movement means simply the moving of the hand or the legs. So in that there will not be any displacement. So movement we can just define, it is the moving of any organ structures. So maybe the moving of your legs, moving of your jaw, the moving of your arm. These all we can bring it under the movement. So I hope it's very clear the difference between the locomotion as well as the movement. Now you just imagine some organisms, the lower level organisms if you are taking, the one structure may do the two functions, both the locomotion and the movement. We can just look into some examples for that. For example, you just take the example of the paramecium. In the case of paramecium, if you're looking, paramecium body, you know that it contains a lot of cilia. So this cilia, if you are taking, they will do the two functions. One, you can see that they will definitely help for movement. The second function that is also very important for the paramecium, that will help for food passage through the cytopharynx. So I hope it's very clear. So the cilia, the structure present in the paramecium, they will help for the movement. Also, they will help for the food passage through the cytopharynx. Similar to that, we can see one more example here. You take the example of the hydra. Hydra, you know that it's a cylindric. You know that cylindrics generally will be have the tentacles in their body. So you take this tentacles. So this tentacles, if you are taking, you can see that they will contain the nidoblast. So with respect to the tentacles, if you are saying, and sorry, with respect to the hydra, if you are saying, this tentacles, the tentacles, this muscular tentacles, they will do two functions. What are the two functions? First of all, without these tentacles, they cannot move from one place to another place. So definitely they help for the movement of the body or the locomotion. So tentacles will first function, they will help for locomotion, and you know that this muscular tentacles are very, very powerful, and also please understand they contain the nidoblast. It's a lot of toxins contained inside of it. So this tentacles also you can say that they will help for capturing the prey. Here. So in this part, what we have seen, the two things only we have seen here. First of all, we define the locomotion. Second thing, we define the movement. The third thing, we have just defined the structures that can be used for the locomotion as well as for the movement. So with respect to the paramecium, if you are taking the cilia, cilia help for the movement as well as for the passage of the food through the cytopharynx. So here, sorry, your movement in the sense should understand it is locomotion. Better word is locomotion. And here help for the food passage through the cytopharynx. So here the tentacles, the help for the locomotion and the help for the capturing the prey. I hope it's very clear to everyone, no doubt. Now, what is next we are going to discuss it is, what are the different types of movements in the human body? That's only we are going to discuss next. So, what are the different types of movements in the human body? So, everybody listen. So, there are different types of movements in our body. Mainly, we are discussing the three types of movements. The first one is amoeboid movement. Second one, we call it as ciliary movement. And third one, we call it as muscular movements. Right. So we'll discuss one by one. First of all, the amoeboid movement. So why the term the amoeboid movement? Because in the amoeba, you know that how they will move. The amoeba will move by streaming the protoplasm. We learned it already in the smaller classes. So while they do this streaming of the protoplasm or the pseudopodic movements, their body will just move up. Similar to that amoeboid movement or the pseudopodic movement, you can observe in our body also. That examples only we are going to look into. So the amoeboid movement will be seen in our body in two areas or in two structures. So the one it is called the macrophages. The other one we call it as leukocytes. So let me explain this part. So why this amoeboid movement is present in the case of macrophages and the leukocytes? It's very important to understand. Because this macrophages and the leukocytes are one of the very important structures for our immune system. Whenever a cell injury happens, into that spot, this leukocytes and the macrophages should have to be reached. So they should have the proper movement, otherwise they cannot reach them. So here what we have to understand, how this macrophages and the leukocytes will get the amoeboid movement. The first part what I said, why they should have the amoeboid movement. So let me give the answer for that. Why macrophages and leukocytes should have this amoeboid movement or pseudopodic movement. Because then only they can reach into the injured side of the cell. So the answer for this, it is then only they can reach into the cell injury site. Now that's clear. Now the next question you have to ask here, it is number one question. I think all of you know what is a macrophage and what is a leukocyte. Generally, the leukocytes means it is WBC. So what is a macrophages? Macrophages are nothing but, you just understand it very clearly that you might be heard it but already, but just give me a brief idea about it. So you know that from the bone marrow only, the WBCs or the leukocytes will be get synthesized. So in the bone marrow it will get synthesized. After that, what will happen to this leukocytes? They will go to the circulation. Okay, the circulation, After spending some time in the circulation, where they will go? They will go to the tissues. Here. So this is tissues. So everybody please understand. The leukocytes, they will be synthesized in the bone marrow. After that, they will be released into the circulation. Then it will be taken into the tissues. Almost all the leukocytes, they will do this particular character. There is no doubt in that. So what you have to understand here, if you take the monocytes alone, once they reach into the tissues, unlike the other leukocytes, the monocytes will spend a lot of time inside the tissues. Usually, the usual leukocytes, if they reach into the tissues, their lifespan will be immediately, it's gone, it's finished up. They will be immediately degenerated up. But unlike to that, if the monocytes reach into the tissues, they will be there for some time. So when the monocyte enters into the tissues, we will no more call it as monocytes, we call it as macrophages. This is the concept behind the macrophages. Here. Now, this macrophage, if you are considered, they have the good phagocytic action. Now the question here it is, how this leukocytes in general and the macrophages specifically, how it will be possible, how this amoeboid movement is possible, that we have to understand. The reason behind why the macrophages and the general leukocytes, the amoeboid movements are possible, it's because of microfilamental structures. So it's because of microfilaments. You might be heard about the cytoskeletons. Cytoskeletons, there are three types. One is called microfilaments. The other one is called it as microtubules. The other one we call it as intermediary filaments. Among the cytoskeleton, the microfilaments only, with the help of this microfilaments only, we are made this macrophages and the leukocytes in general. Now, the question you have to ask is, how the microfilaments will give this particular character? Because microfilaments basically made up of the protein called actin. Because you know that the actin is having the contractile power. So that's the reason you can see that this microfilaments, they will give the contractile power to the macrophages and the leukocytes. Here. So that's the reason we are saying that the leukocytes and the macrophages, they have the contractile power, or they can contract based on the situations. So for example, one contractive movement I can tell you, macrophages, I already said you that they have this macrophages, if you are taking, they have the phagocytic power. How this phagocytic power will be enhanced? It is with the help of this microfilaments. Why the microfilaments can do that? It is because of the actin. Here. And also please remember, the same microfilaments only you can see in the case of microvilli structure also. Here. What is a microvilli? The microvilli are the structure present in the small intestine, help for the absorption of nutrients. Clear? So that also you should remember at this point. Clear? So please understand the microfilaments, two things you may have to remember. One is microvilli are made up of microfilaments, and also please remember the macrophages composition is also happen with the microfilaments, and the microfilaments generally made up of this actin, the contractile protein. Clear, everyone? So because of that reason only, the leukocytes having this amoeboid movement, or the macrophages, and the one leukocyte is called neutrophil, their phagocytic action is can enhance by this microfilament. That also please remember. So the amoeboid movement, I think it's very clear. There is no doubt in this. So if you are very clear about this amoeboid movement, the next thing we have to discuss it is the ciliary movement. So we can look into that. So the next thing we are going to discuss about the ciliary movements. So here I would like to bring your attention. Already we had discussed in the cytoskeleton in the cell unit of life chapter. So that time we have learned it. The cilia and the flagella, they are made up of a cytoskeleton, and its name is called microtubules. And this microtubules, if you are looking, they are made up of the tubulin protein. They are alpha tubulin, beta tubulin, and the gamma tubulin. Here everyone. Now, where and all you can see this ciliary action? That's a question you have to ask me. Ciliary action, where and all we can see it, we have learned many times this. Wherever the ciliary epitheliums are present, that areas you can observe the ciliary action. The number one region, if you are considered, that is trachea. Right. Why the trachea requires this ciliary epithelium? What is the use of this ciliary action in the trachea? Everybody know that. Because our upper respiratory tract is very much exposed with the dust particles and other microbes. So with the help of this ciliary movement, we can remove the dust particles. Here. So that's the use of this particular ciliary movement in the case of trachea. Now, moving on to the next example for the ciliary movement in our body, it is you take the human female body, the female reproductive system. So we have the uterus here, the fallopian tube, then the cervix and the vagina. So here, by the ligament, the ovary is connected. So the two things you have to remember here. One is the specific things about the structure of this uterus. You can see that they have the lateral extension, it's called fallopian tube. And the fallopian tube, this region, we call it as infundibulum. This region, we call it as ampulla. And the proximal to this uterus, we call it as isthmus. You check this throughout this fallopian tube, they contain the ciliary epithelium. You take this infundibulum part and you can also observe this fimbrial region, the finger-like structure. There also you observe it. You can feel this ciliary epithelium. So why the fimbriae and the full fallopian tube contains the ciliary epithelium? We have to clearly understand. The reason behind why the fimbrial part contains the ciliary epithelium, the point is very clear. Because the ovum will be released out from the ovary. What is the one character of the ovum? The ovum released from the ovary, what is the main problem? Their main characteristics, they are immotile. They cannot move. So if they want to move inside this, fallopian tube, the fimbriae line, the ciliary epithelium will help for that. So everybody clear, because they are immotile, so they have to be taken to here. And the sperm will be come here in the ampullary region, ideally, will fuse it. But how they will be taken inside? They cannot move themselves. So this ciliary action will help them to take inside. Clear? That's the one point. So as they are immotile, they cannot move. So ciliary action requires to take inside the fallopian tube. I think it's clear. The second point, you just imagine this, that once the sperm also comes here, fertilization happened and the zygote formed up. The zygote, if they want to move into the final destination, that is the uterus, there also this ciliary epithelium's function will come. So in the female reproductive tract, the two way it will help. One, it is for the movement of the ovum into the fallopian tube. Another one, it is after the zygote formation, zygote will be moved into its final destination, that is the uterus. Ideally, that function also will be regulated and will help by our ciliary epithelium. Points clear? So number one, this. Number two, it is the movement of the zygote. Clear? So that's why someone does not have the proper ciliary epithelium in the fallopian tube, they cannot sometimes conceive. That's the one problem they will face it. So now you might be understand what is the importance of the ciliary movements in our body. You just imagine if the person does not have the ciliary epitheliums in the trachea, it will invite a lot of infections to the respiratory tract. And also you please imagine if a person, the female, does not have the ciliary epithelium in the fallopian tube, sometimes she cannot give the offsprings. So I hope it's very clear that these 2 points. Now finally the muscular movement. So I hope it's both things are very clear. Now finally the muscular movement. So listen, muscular movement, if you're taking, that will go in detail. You take the different or muscle movements all the muscle movements. If you want to move your jaw, if you want to move your legs, if you want to move your arms, everywhere you require the muscle movements. Or else, if you want to do some activities like jumping, so or else if you want to do the running, or else you want to do the skipping. So whatever the things, if you want to do in our body as a movement, the voluntary movements, we require this muscles or the muscular movements. Without that, we cannot make any kind of movements or the locomotion in our body. So it's very important. The muscle movements only we are going to deal in this chapter in more detail. Here, so far we talk about the totally in four concepts. The number one, the locomotion and the movement. Number two, the structures that can be used for the locomotion as well as for the movement in the lower organisms. Number three, the types of movement. And under the types of movements, we have seen all other movements, amoeboid movement, ciliary movement, and the particular muscular movement. Now, in the next part, we are going to deal about this muscles. That's only we are going to discuss in detail, right? So we can look into that. So listen everyone. We have learned already that muscles, if you are taking, they are originating from mesoderm. So if you take the total mass of the human body, it's 40 to 50% is the 40 to 50% is contributed by the muscle tissue only. So 40 to 50% of body mass contributed by muscle tissue. Yeah. So we know that already we learned about it. If you take the muscles, there are different types of muscles. They are the skeletal muscles, smooth muscles, and the cardiac muscles. You take any muscles, whether it is skeleton, whether it is smooth, whether it is cardiac muscles, all these muscles have a general properties. So that general properties only we are going to see next. So all of you please look into that. The next thing we are going to see it is the general properties of the muscles. So the number one property the muscle is having, they will get excited. So they have the excitability. I will explain what is excitability. The second one it is they have the contractibility. So they have the extensibility. And they also have the elasticity. Here. So they have the excitability, contractibility, extensibility, and the elasticity. So you take an example. If any muscle got excited, they will do the contraction, they will do the extension, and they will come back into its normal position. Am I right? So here, what is the exact meaning of excitability? The muscles can excite. So listen, muscles can excite with the help of neurons. We will discuss later about it. Then, once they get the excitation from the neurons, they will contract. So once they will contract, they can extend. Once they extend, after the extension, you can see that they can do like this. So at the time while they extend, the same time we can bring it back into our normal position. So we can say that they are elastic too. Clear? So the general properties of the muscles, they are excitability, contractibility, extensibility, and the elasticity. It's clear? Now, we have to look into the different types of muscles. What are the different types of muscles? We can see that. So how can we classify the muscles? How we will say that there are smooth muscles, the skeletal muscles, and the cardiac muscle, based on what we are saying this. So basic classification we are doing based on their location. The second point we may consider it is the appearance. Third point we may consider it is regulation. Right? So three things we have considered for the classification. Classification points are, so the location, appearance, and the regulation. Now, first of all, we can talk about the first muscle, that is skeletal muscles. So you take the skeletal muscle, what is the location of the skeletal muscles? Skeletal muscles always attached with the bones. So always you can see that a skeletal muscle always will be attached with a bone. You know that this is one bone. This is a humerus. So the skeletal muscles will be attached here. Okay. So all the time, please remember, skeletal muscles will be attached with these bones, right? So that's a one point you can remember. They will be attached with bones. Now, what is their appearance? They have a striated appearance. So who will control them actually? Actually, skeletal muscles will be controlled by our central nervous system. We have the two types of nervous system. One is called, mainly we can divide into central nervous system and the autonomous nervous system. In our case, in our skeletal system or the skeletal muscles, they will be controlled by our central nervous system only, includes the brain and the spinal cord. So the controlling or the regulation by CNS. So what is the advantage if something is controlled by the CNS? That actions we can control it. That means based on our will, we can control the actions. For example, if you want to move something, For example, if you want to move this bone, if you want to move it, this muscle, if you want to move it, at the time when you require, you can do that. Your body will never do this action without you know it. Here. So that means based on your will, you can do that. So that this kind of skeletal muscles, we call it as they are, we can do based on our will, we call it as skeletal muscles are voluntary muscles. Why this? How this voluntary action will be possible? Because they are regulated by CNS. That's a reason. Remember that point. Here, and looking into the other points regarding the skeletal muscles, you have to clearly understand. Skeletal muscles, basically, they are multinucleated. I'm just continuing this part. They are multinucleated. And also, please remember, they are branched. So please remember that point also. We'll discuss more about it in the next part. Now coming into the next, that is the smooth muscles. So where is the location of the smooth muscles? Smooth muscles basically contain in the visceral part or the inside the body, visceral organs, important vital organs. So you can see that this where it will be seen in vital organs. Which are the vital organs? You can see in the lungs, in the kidney region, then GIT, many other vital organ structure you can see that. Okay. So that's why we also call them as visceral muscles. Yeah. And also remember regulation. We have to look into the appearance. Sorry, we have to look into the appearance. Appearance, if you're looking, in the case of skeletal muscles, I told you that they have the striations. So please look into the skeletal muscles. They will have the striations like this. But if you look into the smooth muscles, they will be like this. They have no striations. As they have no striations, they will be very smooth. So that's why we call them as smooth muscles. So you can write it down. No striations. So it's smooth. And from this diagram, it's very clear. If you look on to the smooth muscles, they have the spindle shape. And it's very clear, if you look on to its nucleus, they are the uninucleated. That's clear. So I think this is very clear to everyone. Right. And what you have to remember, the some points while we discuss about the smooth muscles. Smooth muscles, some regions you will see it as circular in shape. Some region you may see it as longitudinal in shape. Some regions you will see the circular, longitudinal and the oblique. Even though their structures are very similar, their appearance sometimes will change it. Some regions it will be the smooth muscles. Let me talk about it. That also. Smooth muscles in different body parts. Smooth muscles in circular shape, you can observe it. What is a good example for that? The very good example for this it is sphincters. You can give the examples of urethral sphincters, then pyloric sphincter, ileocecal sphincter, so like that, all the sphincters you take it, they will be the smooth muscles only, so circular smooth muscles. Now, while we look on to the another one more shape, it is circular and the longitudinal. This is very clear. We have learned already in the digestion part. It is seen in the intestines. It's very popular one, especially in the small intestine, the large intestine regions. You can observe the circular and the longitudinal smooth muscles. Now, in some regions of our body, you can see the circular, longitudinal, and the oblique smooth muscles. That is the two areas. It's very important. One is urinary bladder. Another one it is uterus in the female. Clear? So these are the two regions. You can observe the circular, longitudinal, and the oblique smooth muscles. The circular and longitudinal smooth muscles you can observe in the case of intestines. And the good example for the circular smooth muscles, we call it as sphincters, seen in the urethral, pyloric, and the ileocecal regions. So these are the main points about the smooth muscles. Now, looking into the next one, that is the cardiac muscles, we have to see that. So cardiac muscles, while you're looking, you will have to understand many things. Right. Right. So first of all, the location part. Location, if you consider, you can see that the cardiac muscles will be seen in the heart. No doubt in that. And while we're looking onto the striations, that's part you should be little more clarity you should be having. First of all, you have to understand why this striation is actually in the case of skeletal muscles. These striations in the skeletal muscles are nothing but the actin and the myosin filaments only, no doubt in that. But if the same actin and the myosin filaments are present in the cardiac muscles also, but the density of that will be comparatively less. Here, so that you can see that in the case of cardiac muscle, it will be having the faded striations. The faded striations. Remember that point. Now look into the regulations. That is also very important. You know that the cardiac muscles, you can't do anything. It's not in your control. So remember the point, that's a very important point. The cardiac muscle generally will be controlled by the autonomic nervous system. So here, what you have to understand together with the this cardiac muscle and the smooth muscle, I would like to say that together. If you consider the cardiac muscle, it is controlled by autonomic nervous system. And also you take the smooth muscle, they are also controlled by autonomic nervous system. So as they are controlled by the autonomic nervous system, both these muscles are involuntary in action. You cannot control. Smooth muscle is also involuntary. No doubt in that. Clear? So I think that point is very clear. Now, look into the next one. That's really important. So from the look itself, from the appearance itself, you can say that it is branched. Right. But you see this cardiac muscles, basically they are uninucleated. But you just imagine cardiac muscles all the time, they have to work. Without the heartbeat, you cannot survive for more time. So the heart has to be beat all the time. But what is the one problem when we compare with this skeletal muscles? Skeletal muscles are basically multinucleated. Okay, we have written here. But this one is uninucleated. But even though they are uninucleated, how they will overcome that problem? It's really important. So if the cardiac muscles want to overcome that problem, the creator had designed a special thing, and that is called intercalated disc. So how the intercalated disc will overcome the situation, or how they will help the cardiac muscle to overcome the situation? It's very simple. Because if you take an intercalated disc, so let me write about it. If you take an intercalated discs basically are, they are gap junctions only, right? So basically intercalated discs are gap junctions only. So gap junctions, what is the good thing in the gap junctions? The ions can easily, they can dance and pass here. So that may not be a problem. They can easily pass their information from one region to the depolarization and everything from one region to another region. So they can work as a one unit. Because multicellularity in the muscles, the main intention behind it is they should be work as a one unit. For example, your heartbeat, when it happened, your entire heart, almost every heart part will get beaten up. You just move a skeletal muscles, your entire the skeletal muscles will do this work. So you cannot ask your skeletal muscle, okay, skeletal muscle, you don't move it, I don't want to move you. You can't do that. So please understand, if they want to work as one unit, skeletal muscles require the multinucleated, but that problem, even though they have only the uninucleated in the case of cardiac muscle, but they will, how they will overcome that? By the intercalated discs. Intercalated discs are the contain the gap junctions. Clear, everybody? It's very clear. No confusion in this also. Absolutely clear. So if you are very clear with this part, now we can endorse into the skeletal muscle. So in this chapter, we will go more details about the skeletal muscles. So we can look into that. Yeah. ஒரு நிமிஷம் பார்த்துட்டு சார். ஓகே சார். So we can restart. The skeletal muscles, they contain a lot of this muscle bundles. That muscle bundles also you can call it as vesicles. So inside the muscle bundles, you can see the muscle fibers. Muscle bundles also. And you can see this muscles, they will be covered by a covering. It's called epimysium. And you can see that all these muscle fibers, they will be covered by another covering. It's called perimysium. And inside the muscle bundles, there is another covering. It's called endomysium. Right. So you can see that what are the three layers we have seen? The epimysium, the perimysium, and the endomysium. So epimysium, what is epimysium? The entire muscle will be covered by epimysium. This muscle fibers altogether, they will be covered by a covering called perimysium. And inside the muscle fibers, you can see that they will be covered, inside of it will be covered by another structure called endomysium. Clear all these things? Now, if you take this muscle fibers, if you dissect the muscle fibers, you can see many other structures. Okay, you take a muscle fiber. This is the one muscle fiber. Inside the muscle fiber, if you are taking, you can see the another things, and it's called myofibrils, or we can call it as myofilaments. Right. So inside the muscle fiber, you can see the myofibrils or the myofilaments. So if you consider this myofibrils, or myofilaments, there are two types of myofilaments. One is called actin, another one is called myosin. Actually, this myofilaments, they're designing only actually giving the striation to the skeletal muscles as well as the cardiac muscles. Clear? So please understand, they are that myofilaments are actin and the myosin only. Clear? Now come back to this structure. Listen, if you take the two muscle bundles, they are connected each other. So this structure, we call it as fascia. Right. So the two muscle bundles are connected each other by a structure called a fascia. Fascia, if you're looking, the fascias are connective tissues. So this inflammations to the fascia is very common. It's called fasciitis. That's clear. Now moving on to the next, we have to go more details about this muscle myofibrils or the myofilaments. So listen, if you open a muscle fiber, you can see that if you take just open a muscle fiber is having a membrane. So I'm just talking about the muscle fiber structures. So this muscle fiber, if you're looking, its membrane, the muscle fiber membrane, we call it as sarcolemma. Okay, so inside of it, we'll have the cytoplasm, and that is called sarcoplasm. And in this, you can observe endoplasmic reticulum, this endoplasmic reticulum present in this muscle fibers, we call it as sarcoplasmic reticulum. Clear that also? Inside the sarcoplasmic reticulum, if you are observing, you can see the calcium ions. Right. Now, listen to the next point. That diagram I would like to draw here also, because to make you understand little more things. Now listen, you might be observe what is the particularity of this membrane. So this membrane, they invaginate into little more deeper. Actually, because of this characteristic structure, they have a T shape. Okay, because they have this T shape like this. This particular muscle fiber sarcolemma structure, we call it as T tubular system. So because of this characteristic invagination with a T shape, we call this sarcolemma, and this together structure we call it as T tubular system. Especially this sarcolemma part, the membranous invagination, we call them as T tubular system. In this T tubular system, in this invagination part, if you're looking, I already explained there is a sarcoplasmic reticulum, right? And if you look on to this T tubular invagination, you can observe many other things. This region you can observe a receptor, and this receptor we call it as dihydropyridine receptors. This dihydropyridine receptors, they contain calcium channels. There is another one more receptor here that is present in the sarcoplasmic reticulum. This is sarcoplasmic reticulum, and this is called as, listen, this one we call it as ryanodine receptors. Ryanodine receptor, please understand, they also contain the calcium channels. Right. I hope it's very clear. Now, if you look on to the sarcoplasmic reticulum membranous part, there you can observe another one enzyme, and that enzyme we call it as calcium Q plus ATPase. Clear? So these are the points you may have to remember about this sarcolemma. Inside of it, the cytoplasm, it's called sarcoplasm. Then inside of it, endoplasmic reticulum is there. We call it as sarcoplasmic reticulum. Inside of it contain the calcium 2 plus ions. The characteristic, this muscle fibers, its sarcolemma part, we call it as T-tubular system. And you can also observe the two types of receptors. One is ryanodine receptor, another one is the dihydropyridine receptors. Both contains the calcium channels. And the sarcoplasmic reticulum, you can see that there is an enzyme. It's called calcium 2 plus ATPase. These points are very important, but I would like to add one very simple point. The sarcoplasmic reticulum is the modified version of, you know that we learned it before, the modified version of smooth endoplasmic reticulum. Remember that point. That's too important to remember. So I think that parts are very clear. Now, inside this T-tubular system only, you can see that this myofilaments will be present. Now we are going to discuss about it. So generally speaking, listen, if you consider the actin, generally speaking, it is, we call it as I band. Because actin is having a property of isotropic. They are generally isotropic. Because why we call it as isotropic? You just imagine, you take the actin filaments, the light is passing through that. The light will not be scattered too much in the case of actin filaments. What is the point you may have to remember? They will not scatter too much. So as the light is not scattered too much, we call them as isotropic. But you take the example of the myosin. Myosin is not similar to that. Myosin, if you consider, we call them as A band. What's the reason? Because you take this as the myosin. When the light passes through that, they will be scattered too much. Because of that reason, myosin we call it as anisotropic. And actin, already I said, it is called isotropic. Clear? So absolutely clear. So no confusion here. Right. Now what we have to understand, you consider this actin and the myosin. This actin and the myosin, they are the contractile proteins of the muscles. So this actin and the myosin, if they move in the certain pattern only, the contraction will be bring it out. So in the first part of the next lecture, what we are going to see it is how this actin and the myosin have arranged. Number one. Number two, we will see that the structure, the detailed structure of this actin and the myosin. In the number three part, we will see that how this actin and myosin will bring the muscle contraction. So it includes three parts. So we can take one by one. So listen everyone to the next part. So these parts are very clear. So I'm going to rub this. So first of all, we can see how the actin and this myosin, they have arranged it. Right. If you take the myofilaments, this actin and the myosin, how they have arranged it. So we already said that the actin is I band and the myosin is A band. So this is A band and this is I band. So this is I band. Okay, again, this is A band. This is I band. Right. So this is I band. This is also I band. This is I band. This is also I band. First of all, we will look into the structure. So this is myosin, this is actin, you know that. If you look on to the I band, in the both side, you can see that I band is bisected by a line, and this line we call it as Z line. Okay. And if you look at, you can see very clearly the A band is overlapped by the I band. This is the A band, it is overlapped by the I band. But this region, this part, you can see that it is not overlapped. So where the A band is not overlapped by the I band, that region we call it as axon. So let me write about it. So what is the axon? So I marked it. This is the axon. This is the axon. So where A band is not overlapped by I band, that zone is called axon, right? Now, if you take the center of the axon, there is another line, and that line we call it as M line. So please make sure that you understood better. The center of the M line, center of the axon is called M line. Clear? No doubt there also, absolutely clear. Now, listen. You know that this I band and the A band, their particular movements or their relationship only actually will bring the muscle contraction. So we can see what will happen during the time of a muscle contraction. So during a contraction, I would like to draw another diagram here. So this is the A band. Right. So look into the diagram. You can see one thing. During the time of contraction, you can see that this axon is get disappeared. That means the A band is fully overlapped by the I band. So let me make it very clear. You can see that the position of the I band, if you are considered, during the time of muscle contraction, they will be close to each other. Am I right? So that who will be disappeared during the muscle contraction? During the time of muscle contraction, listen, that point is really important. The axon will get disappear. And you can also observe that this I band length will change. Right? That is also one point you can remember. So one is axon is disappeared. Second point is I band length will change it. But one thing you can observe here, it is the position of the A band. They have no change at all. So there is no change in the A band in the length during the time of muscle contraction. So that's also very important. No change in A band. Clear? And the one more very important point you consider, we have very clearly understand this from the diagrams itself. The distance between the Z line, it became less. So during the time of muscle contraction, distance between Z lines will be reduced. So we can make it a sense like this. This A band and the I band, their relationship that will bring the muscle contractions. And during the time of muscle contractions, I band length will change. But all these length increase and the decrease process happening in between the two Z lines. Am I right? In between the two Z lines. So as it is happening in between the two Z lines, we can consider the distance between the two Z lines, that is the one functional unit of the muscle fibers. Here, just imagine, because the distance between the two Z lines only actually will bring the contraction. So many this kind of distance between the Z lines will bring the one contractions. So we can say that the distance between the two Z lines, that is the contractile unit of a muscles. What is that? Contractile unit of the muscles. So this distance between the two Z lines, generally we call it as sarcomere. What do we call it as? The distance between two Z lines, what we call? Sarcomere. This sarcomere, you can call it as they are the contractile unit of a muscle. You can also call it as they are the functional unit of the muscle fibers. So sarcomeres, too many titles we can give it. They are the functional unit of muscle fiber. They are the contractile unit of the muscle. Okay, that's clear. So absolutely no doubt. Now, look into the next point. Actually, whatever the contraction happened here in the this contractile unit in the sarcomere, that force has to be come up. See, if you punch someone with a less force, and if you punch them with a more force, the force getting into the, or the punch getting into the intensity of the punch will be different. Am I right? So the point here you have to understand, if I punch someone like this, or if I punch very intensively, the punch force will be different. So actually, this contractile unit, the sarcomere unit, force if you want to give outside, the force of the contraction if you want to give into extracellular region or extracellular or outside this muscle fiber, we require a protein, and that protein name is called dystrophin. So listen, everybody listen. What is the importance of a protein called dystrophin? Let me speak about it. Whatever the contractile force is bringing by this sarcomere, If you want to give it extracellularly, that means outside the muscle fibers, we require a protein called dystrophin. So let me write about the dystrophin. So what is the function of the dystrophin? The force of sarcomere or contractile unit want to give extracellularly we requires dystrophin. So what is why I discuss this point? There is an importance. What is the importance of that? Because in the last part of this chapter, you will learn one thing. There is a disorder called muscular dystrophy. So actually, muscular dystrophy means what? It's nothing but a deficiency in the dystrophin. It's basically a genetical disorder. Now you can easily say that if the dystrophin is absent or deficient, what will be happen? No force. So you not be have the no muscle actions or the no muscle force. So the very characteristic symptom of this will be the muscle weakness. Patient cannot extend the life too much. Maybe he may go up to the 35, 40 maximum in maximum. Many of the patient will be die in the middle of 20s. So that's the one part you may have to remember. So I think this is very clear to everyone. There is no question. Right. If you're very clear with us, now what we have to discuss, we have to discuss about the detailed structure of actin and the myosin. So listen, everyone. So first of all, we can consider the actin. Actin, if you consider, they are made up of filamentous protein is called F actins. No doubt. So the filament is, that's the reason we call it as F actin. So you consider this F actins. F actin is actually a polymer. Its monomer unit, we call it as, the monomer of this F actin is G actin. Clear? And if you look on to this actin, you can see that, you will look into that. They are helically arranged. I'll draw the diagram here. So I'm going to rub this part. So actin, I think this much part is very clear. I'm going to rub this. So I will write up there itself. So actin is made up of F actins. Right. So all of you listen. So what we have seen, it is made up of F actins. Right. So its monomer, we call it as F actins monomer. Is G actin. Right. So that much clear. Now you look onto the actin, this structure exactly. They are helically arranged. Right. And in the grooves of this actin filaments, you can observe another one protein. And this we call it as, in the intervals, you can see that. And this we call it as tropomyosin. Right, everyone? Understood that point also? Yeah. So the tropomyosin, so this is the actin, this is the F actin. So they are helically arranged. This is the one helical arrangement. That means this is a one line of one thread you can see here, one thread here, and the two F actins will be seen in the one actin filaments. So they are helically arranged. And just over that, we can see another protein. It's called tropomyosin. Now the next one is really important. And very associated with this tropomyosin and this actin, you can observe another one protein, you can see it, and that's called troponin. Right. So the actin filaments, you can see the tropomyosin, the troponin also. So here, this is very important, actually very important thing it is. So let me write it out. So in those actin filaments, what are things you can see? First thing, what you saw it as tropomyosin. The second thing, it is troponin. This is really important. Troponin contains actually three different proteins. We call them as troponin I, troponin T, and troponin C. What are that? Troponin I, troponin T, and the troponin C. So why the term troponin I? So listen that point very clearly. Right. So why the term I? Because this I, troponin I part, they will inhibit the association of actin and myosin. That's why we call them as troponin I because of the word inhibit. Now look on to the troponin T. Troponin's T part, or the T, that particular protein, they have attached with tropomyosin. And finally to the troponin C, that's also really important. Bind with calcium. That's the reason we call it as, it's a troponin C. So troponin ITC, it's very clear, no doubt. Now, if you are very clear about this, now we can talk about the myosin part. So we already learned that they are anisotropic, and comparatively they are more thicker compared to the actin. So we call them as thick filament. So with respect to the myosin, actin is a thin one. We call it as thin filament. But you never forget, the thin filaments is composed with these three proteins. It's not only because not only with this actin. Remember that point very clearly. So this is the thick filament. Now let me look into the next, the structural part. This is the one, myosin. Just look into that. Myosin is having two parts. You can also say it as three parts. This lengthy part, you can call it as tail. And this part, we call it as hinge. And final, this part, you call it as head. Right. Similar to the actins, we talk about F actins, myosin is a polymer only. What is the monomer of the myosin? Monomer of myosin is, it's called maromyosin. It's called maromyosin. So that's clear. Now, if you look into this maromyosin present in the tail part, they are comparatively lighter in density. Their molecular weight is comparatively less. So we call this tail part maromyosin. Generally, we call it as they have the less molecular weight. We call it as light maromyosin. But this head part, the head plus hinge part, you consider, they contain that maromyosins are their density is more, their molecular weight is more. So generally you call them as heavy maromyosin. So light maromyosin and the heavy maromyosin. So what we can tail we will denote with light maromyosin. The head we denote with heavy maromyosin. So you can call it as respectively LMM and HMM. Clear? Point is very clear. So the heavy Meromyosin and the light Meromyosin. So I think it's very clear. This is light and this part is heavy. Now the very important next point. So you just look onto this diagram. This hinge part, and you can see that this head part together, they will make just a cross arm appearance. So this hinge part, from this hinge part, you can see that they will make an angle like this, and this side also they will make an angle like this. So as they are angle make like this, like a cross arm, this structure we call it as cross arm. So how this cross arm will come up? From the hinge region to the head region. That together structure we call it as cross arm. Now look on to the next point that is also very important. If you look into this myosin head, there you can observe two attachments or two things you can observe there. The one region inside this myosin head, the one for the actin attachment. The actin will be attached here during the time of contraction. And this one head, the head one part, that is for ATP binding. Clear? The one head's one part is for the actin attachment or the actin binding. The another part for the ATP binding. So listen, that point is very important. The next one, where the ATP binding will happen, there you can see a special type of enzyme. It's called ATPase. ATPase will generally do what? They will break down the ATP. They will break down the ATP into ADP plus inorganic phosphate. Clear? That is also clear to everyone? No confusion there also? Right. Now listen, I would like to make a very important point here. That's a usual question they will ask it. What is a cross bridge and a cross arm? So make it very clear this. Cross arm, already we learned it. What is that? The hinge part but plus the head part, they will join together, they will form the cross arms. But what is a cross bridge? So you know that, you just imagine this is the actin filament and this is the myosin filament, the myosin head. So you can see that during the non-contractile peak, there is a gap between the actin filament and the myosin filament. But during contraction, what will happen? During the contraction, that gap will be get bridged up. That we'll learn later how that will happen. But during the time of contraction, this gap will get bridged up. There will be a bridging of the gap. So that's why this structure together we call it as cross bridge. Clear, everyone? Understood this? So the cross arm versus the cross bridge, I think it's very clear what is a cross arm and what is a cross bridge. Clear? So the cross bridge what will happen during the time of contraction? The gap between the actin and this myosin head will be bridged up. That's why we call them as cross bridges. Clear that point? Now, the next what we are going to see it is how a contraction will happen. That's a point we are going to discuss. So the next point we are going to discuss, it is muscle contraction. So listen everyone, I told you one thing in the beginning regarding the excitation. What I said, without a neuron, a muscle cannot excite. So every muscle contraction, what we require it is, we require the help of a neuron. So that's neuron, what kind of neuron it should be? It should be a motor neuron. What is a motor neuron? Motor neurons only generally will cause muscle contractions. That will detailedly discuss in the neural control and coordination chapter. But let me make it very clear. So I'm going to discuss about the muscle contraction. So listen everyone. So what I said, the muscle contraction, if you want to bring it, what you require? You require the help of a motor neuron. So first of all, we can start from that. So listen everyone. So first let me make a motor neuron. So motor neuron is having the dendrites. They receive the sensory information. So they are passing the sensory information. So this motor neurons will be covered by myelin sheath, right? So here you can see the knots of Ranvier. But now listen, in the terminal part of the axon, so let me make it beautiful. So if you look into the terminal part of the axon, there will not be any myelin sheath. Remember, terminal part of the motor neurons, there will not be any myelin sheath. So what is the next thing will happen? They will start to make the branches, right? Here. And you can see that in this terminal part, if you're looking, these axons will be covered by another covering. And it's a kind of glial cells. We call it as teoglia. It's a kind of squamous cells only. Now, after that, what will happen? Listen. In the end part of this axon, you can see a vesicles. So this is called synaptic vesicles. So listen everyone, the synaptic vesicles, they are not very straight structure. You can see some kind of invigilation here. All right. Right. So this region, this is called synaptic vesicles. Sorry, synaptic node. So inside the synaptic node, you can see that vesicles, so many vesicles. So inside these vesicles, you can observe the acetylcholine. Right. That's clear. So synaptic nerve, acetylcholine is very clear. So this is called vesicles. So this structure is called vesicles. Now, this region, if you are taking of the synaptic nerve, and that we call it as synaptic cleft. Now, listen to the next point. That's an important part. So, listen, everyone. So, the muscle contraction is the first requirement we have fulfilled. That is a motor neuron. This is a motor neuron. So, this motor neuron, how they will bring the information? So, first the dendrites will receive the information. It will pass through the cell bodies. Then what will happen here? Here the action potentials will be created. Action potentials will bring the this impulses to here in the terminal part of the axons. So here point you have to understand, many points you have to understand here. This action potential or the depolarization process or the action potential, I will talk as action potential. This action potentialities keep on coming into this from this this dendrites, then into the cell body, then into the axon. And if the muscle contraction, if you want to bring it, it has to be passed through the muscles also. But how they will pass it? That's very important. Now listen to the point. When the depolarization happened to the axons, depolarization happened to the axons, or we can say that depolarization happened to the motor neurons. All right. So the depolarization happened to the motor neurons. Okay. Just imagine depolarization happened to the motor neurons. When this depolarization happened in this axons, the one thing will happen. This depolarization will bring calcium influx. That means calcium will be moved inside the axonal terminal. Calcium move inside the axonal terminal. Clear? What is the requirement of this calcium influx? Because this vesicles, if you are taking, the vesicles for the acetylcholine, if you are taking, they are calcium mediated. So when the calcium influx happen to the axonal terminal, what will be happen? What will be happen? You can see that only this part I will take it out. So these are the vesicles inside of it, acetylcholine. Now listen, when the more amount of the calcium come inside, this calcium will be come and bind here because so many calcium receptors are present. When the calcium bind here, this vesicles will get opened. Clear? So listen, calcium influx happen, calcium moves inside the axonal terminal. After that, what they will do? Calcium bind with receptors of acetylcholine. While they bind with the acetylcholine receptors, what will be happening? Immediately acetylcholine will not be released. They have to do one more thing. What is that? This vesicles contain a protein, and this axonal membrane also contain a protein. They also should be joined together. This is the one protein. They have to join together. Right. So the vesicular protein, we call it as synaptobrevin, and other one we call it as syntaxin. So listen, when the calcium bind with the receptors of or the vesicles of this acetylcholine, it will be slowly opened up. Apart from that, this vesicle contains a protein called synaptobrevin, and the axonal terminal contains, this is the synaptobrevin. The axonal terminal contains another protein, it's called syntaxin. They also will make a connection like this. So by the time only who will be fully open? Our vesicles will be open. Once the vesicles opens, the acetylcholine will be come out. Clear? So they will be reaching to what part? They will be reaching to the synaptic cleft region. Clear? That point is very clear. So we understood that acetylcholine released into the synaptic cleft. Now, what is a acetylcholine? You know that it is a chemical messenger or it's a neurotransmitter, especially for the muscles. So next, what should be happen? It has to be passed to the message has to be passed through the muscles. How that will be happen? That only we are going to see it. So listen. So in the next thing it is, you have to understand the muscles also will be joined with this neuron. Actually, which part of the muscle will be joined with? The sarcolemma part. Clear? Because sarcolemma is the membrane, this is also another membrane, so that this membrane and this membrane, they will make a synapse there. So please understand, if you want to bring the information from a neuron into the muscles, you have to understand one thing, some basic things. First of all, we require a axon. That axon should be having a axonal terminal. That axonal terminal, we should be have the vesicles inside of it, acetylcholine. And this axonal terminal part will be connected with a sarcolemma, that is the membrane of the muscle fibers. Clear that much? Now, you please understand. If you consider the axon of the motor neuron, this is the axon of the motor neuron, plus sarcolemma of the muscle fiber, their only actual information or the messengering process is happening. So we will call it as this one axon of motor neuron plus sarcolemma of the muscle fiber. Together we call it as one motor unit. Clear? Okay, clear? And also please remember, where the sarcolemma and this axonal terminal, they will join together, they will meet each other, and that junction we call it as neuromuscular junction. So please write down. So everybody, the junction where axonal terminal and sarcolemma meet each other is called neuromuscular junction. Clear? Absolutely clear to everyone? So this is a sarcolemma. Now, please understand very clearly the next point. So let me make it very clear with one more diagram. So this is the axonal terminal. So here comes this is our synaptic cleft region. So this is our vesicles, inside of it, the acetylcholine. Right. So acetylcholine release up to this one. Now listen to the next point. This is our sarcolemma. Right. So this junction, we call it as neuromuscular junction. Okay, together we call it as a motor unit. Now the next point you have to really understand. So this is acetylcholine. They reach into the synaptic cleft. Now listen, this is vesicles, no doubt in that. And we also talk about the synaptocin and the synaptobrevi. Now listen. The next point you have to really understand. When this acetylcholine, so these ports are acetylcholine. So acetylcholine, you have to really understand how they will pass the information, how they will act as the chemical messengers. For that, what you have to understand, if you're taking the sarcolemma part, there contains so many sodium channels. Right? So many sodium channels they contain. But the sodium channels, what is the importance and what is the character of the sodium channels? The sodium channels are ligand gated. What is that called? Ligand gated. What is the meaning of ligand gated? Ligand gated means These sodium channels are controlled by the chemicals. So who is actually the chemical here? It is nothing but our acetylcholine. Acetylcholine will come and bind here. Then only the sodium channels will open up. Here, this acetylcholine will come and bind in the sodium channels. Once the acetylcholine, so these are acetylcholines. So all these are acetylcholines. When the acetylcholine bind the sodium channels, as it is ligand gated, acetylcholine gated channels, the sodium will be start to release. When the sodium start to release, please understand that will bring depolarization. Clear? Everybody, make it very clear. So when the acetylcholine come and bind in the sodium channels in the sarcolemma, they will bring the depolarization. Now, you just look into the next part. It's also very important. Let me make it very clear. This sodium channels, the sodium just released out, depolarization comes. Now listen to the next point. Listen. Now let me make it a little more enlarged. So this is sodium channels. All right. The sodium channels bind with acetylcholine. Right. So depolarization comes up. Once this depolarization, first, listen very carefully. This will stimulate they will stimulate the dihydropyridine receptors. We already learned about it. You can go back and see the diagram. Dihydropyridine receptors. Right. Now, their stimulation, it will stimulate the next one, and that we call it as ryanodine receptors. So first of all, the depolarization first, what they will do, they will stimulate this dihydropyridine receptors and then the ryanodine receptors. What will happen now? By the time sarcoplasmic reticulum, they will release the calcium into the cytoplasm or the sarcoplasm. Clear? So listen, everybody make it very clear. Let me make it once more. So I would like to write here. So listen. Right. So listen, everyone. So first, what happened? The motor neuron, from the motor neuron, what happened? Acetylcholine released, bind with sodium channels in sarcolemma. So after that, what happened? Depolarization. That depolarization, what happened? What depolarization? Stimulate dihydropyridine receptors. Dihydropyridine receptor. I will just write as DHP receptors that stimulate the ryanodine receptors. and that cause calcium release into sarcoplasmic reticulum. Clear this much? It's very clear. Now, once the calcium release into the sarcoplasmic reticulum, now we can imagine what will happen. In the sarcoplasm, what is present? What is present? Or what is present here? The sarcoplasm only we have the our actin. So in the actin, what we have it? We have the tropomyosin as well as the troponin. So this is tropomyosin and this is troponin. So what will happen? So listen, everybody listen here. When this calcium release into the sarcoplasm, immediately what they will do? Calcium release into sarcoplasm. What will happen? Because the first thing will happen, this calcium bind with troponin C. Then what will happen? Once the calcium bind with the troponin C, by this action, please understand that the mass between actin and myosin will be removed. The mass between it was mass, that mass will be removed. That means actin and myosin bind each other. Clear? And so the muscle contraction will happen. Clear? So let me make it very clear, the points very, very clearly. So the first motor neuron is coming, the acetylcholine released. They will bind with the sodium channels in the sarcolemma. Depolarization happened, stimulate the DHP receptor, stimulate the ryanodine receptor, calcium release into the sarcoplasmic reticulum. That calcium will bind with the troponin C. Troponin, while they attached up, the mass between the actin and the myosin will be removed. So the actin and the myosin will be bind each other. So the muscle contraction happens. Clear? Now, let me make it clear to the next point, because these diagrams are very important. So I would like to add one more point here. So diagrammatically, I want to tell you everything again, because that will be very helpful. So first of all, an axon is coming. Okay, they are bringing this particular depolarization. So acetylcholine is here, right? So acetylcholine vesicles, they have the calcium receptors. Right. Now, what is the next thing? There is a region called synaptic cleft. Right. Now, there is another structure here. It's called sarcolemma. Right. So here we have sarcoplasmic reticulum. Right. So here we have DHP. Here we have ryanodine receptors. Here we have calcium 2 plus ATPase. All right. So I will just write down every point what I said very clearly. Impulse from motor neuron, okay, cause calcium influx in axonal terminal, calcium bind with vesicles, acetylcholine released into synaptic cleft. Okay, this acetylcholine bind with ligand gated sodium channels in sarcolemma. I already discussed that. This is the right. Okay, ligand gated sodium channels. So that will cause sodium leakage, depolarization, that will stimulate the ryanodine receptors, that will stimulate. So first stimulate the DHP, then ryanodine. Then that will stimulate the sarcoplasmic reticulum and release of the calcium into sarcoplasm. Okay. This calcium bind with troponin C and finally that will unmask the actin myosin binding site. So that contraction. Clear? So everybody make it very clear this. Now what we have to understand, one point here it is. Very clearly everybody listen. So this is about the muscle contraction. Now what will happen during the time of muscle relaxation? So this is very important, as same as muscle contraction. So during the time of muscle relaxation, listen very carefully this. From the sarcoplasm, calcium will be taken back into the sarcoplasmic reticulum. The calcium will be taken back. How this calcium will be taken back into the sarcoplasmic reticulum? With the help of this calcium 2 plus ATPase. So remember, during the time of relaxation, calcium will be taken back into the sarcoplasmic reticulum from the sarcoplasm with the help of this calcium 2 plus ATPase. Let me make it very clear. During relaxation, calcium will be taken back to sarcoplasmic reticulum with the help of calcium 2 plus ATPase. Right. Once this happens, calcium level decrease in sarcoplasm. So that what will happen? Troponin C and calcium cannot attach. Cannot. The troponin C and the calcium cannot bind. If they are not binding, what will happen? Again, it will mask the actin and the myosin binding site. Clear? So this is a point you may have to remember. So there is a muscle contraction. So similar to that, there is a muscle relaxation. So muscle relaxation, this particular character only I have told here, the mechanism only I told here. Clear? I hope it's very clear. So now you understood how the I band and the A band, they will work during the time of muscle contraction. How this inside the motor neuron, how the information will pass into the sarcolemma, you understand. Now, what we have to understand, the most important part is coming up. In the molecular level, what is happening here? Because you know that the very important source of energy for the muscles, it is ATP. How this ATP utilization will be happen in the muscles, that also we have to understand. Then only this muscle contraction will be completed. So let me make it that also very clear. So everybody listen here. So we know very clearly. So listen everyone. This is the actin filament, you can imagine. Right. So there is a myosin. Right. So listen very clearly this. So this is the actin. And this is myosin. I don't have to say that, it's myosin. Now there is a binding site for the ATP as well as the actin. So listen. And I told you very clearly, this is the ATP binding site and this is the actin binding site. So listen, when the ATP binds us, this region with the help of ATPase. What will happen? They become ADP plus PI. So just imagine this ATP have just binded here. So what will happen? They become energized, the ADP plus PI. The energy will start to release. So here we have our sarcomere. So the Z line is the sarcomere part. Do not forget about this Z line, right? Now what will happen? Now listen everyone. So first of all, the ATP bind here. ATP just broken down into the ADP plus PI. Now, when the metabolism further gone up, what will happen? We can see that. Listen, this is actin, right? This is okay. So listen, when this metabolism is further gone up, it is little more energized. So what will happen? Our myosin head will be go up like this. It is just in the 90-degree angle, right? So here also you can see that this is ADP plus PI. Now, after that, so by the time also please remember our sarcomere is there, the Z line is present there. It's almost in the same distance. Now look on to the next one. The next is really important. While it is metabolization of this ATP further happened, listen, the maximum energy utilization will come. So what will be happen? We can see that. So this is actin. This is actin. Okay. Now what will happen? They will utilize the maximum energy so that this myosin head will go into 45-degree angle. Here. And also please remember, now the contraction in maximum, so you can see that this Z line will become very closer, right? Here, so here it is ADP plus PI, right? Now listen to the next point. Here still ATPs. This is the enzyme ATPase. That is the ATP binding site. So now you can see that. Let me make it very clear. During the time of muscle contraction, ATP will be bind in the myosin head. In the ATP binding site, what is present? ATPase is present, so that it will bring the, it will metabolize the ADP plus PI or the inorganic phosphate. So in the first diagram, I just shown that the ATP just bind. Okay. In the second diagram, the ATP, they just broken down into ADP plus PI and they start to get the energy. They got the energy so that this myosin head attached with the actin filament. So they are in the 90-degree angle. Now here maximum energy they have utilized, so they gone into the 45-degree angle. And you can also see that this Z line, they came closer. Now please understand. Now the next point it is, this is now they are in the contractile state. Now, if you want to release out this contractile state, one thing only we can do it. What we have to do it is, this energy state, how can we just broken? It's very simple. The simple mechanism it is, what is a simple mechanism? They will come into the normalized state. It's very, very simple. ATP again will bind it. Here. So if the ATP again bind here, the actin and the myosin head detachment will happen. That means this ADP and the PI, that energy state level will be released out and they will be coming to this state. So again, they will go to this state, then that cycle will be get repeated. So here ATP binding happened. Here, so listen, you can see that the first, the actin you take, the myosin you take, they have uh, just attached. That means ATP is just act on it. Further, they broken down and start to get the energy. Now in this phase, maximum energy, so the maximum contraction will happen at this stage. So if you want to just release out this contractile state, you have the another ATP should be come there, a new ATP should be come up. Am I right? So without bringing this new ATP, you cannot release them out. You cannot remove the cross bridge mechanism. Am I right? So just look on to this. Now they have made the cross bridge. Here. Now they are made the cross bridge because they just remove the gap between. So they are in the 45-degree angle like this. So the cross bridge have formed up. So this cross bridge, if you want to remove it, what we should bring it? We should bring the new ATP. Clear? So what is the one clinical application here or the applied physiology here? You know that when a person die, what will happen? Automatically ATP will start to decrease. So after the death, if the new ATPs are not coming, what will happen? At the time of a death, a patient was like this. But the patient died and no more ATP. So what will happen? Patient's hand will be like this only. If you are not keep the patient's hand the proper way, immediately after the death, when the time progress, this hand, you cannot further make it like this in the normal position. The reason behind it is this, you cannot release this contracting or this, you cannot remove this cross bridge mechanism when the time progress. So that you can see one characteristic feature in the case of dead bodies. After the death means around 3 hours to 12 hours of the death. Immediately after the death, you can't see that. Around 3 hours, the stiffening of the body will start because of this decreased ATP. And it may last up to 12 hours. After that, the body start to degenerate, so you cannot observe. The initial 3 hours, you cannot see it. But after the 3 hours, this stiffening of the body by the lack of ATP will be very visible. And that will be last up to 12 hours. After that, body will start to get degenerate. We also call it as putrefactions. So please remember, this lack of ATP in the death time will cause the stiffening of the body. And that condition, we call it as rigor mortis. This rigor mortis will last for 3 hours to 12 hours. What is a good thing about the rigor mortis with respect to a police officer or a forensic surgeon? He will be able to understand the position of the death, and he can also understand the time of death. These two things he can easily understand through this rigor mortis mechanism. That's a one point you may have to clearly understand. So rigor mortis is the one point you may have to clearly understand. So I hope it's very clear that also. Now we can move on to the next part. So look into the next one. So the next thing what we have to understand, what are the two types of muscle fibers? There are two types of muscle fibers. One is red muscle fiber. Another one we call it as white muscle fibers. If you consider the red muscle fibers, why they call it as red? Because they will contain more myoglobin, more mitochondria, right? And they contain less sarcoplasmic reticulum. So in this case, the white muscle fibers, the myoglobin will be less, mitochondria will be less, but sarcoplasmic reticulum will be more. Here, that point also please remember. Now, after that, while we're discussing about the skeletal system, we have two types of skeletal system. One is called axial skeletal system. The other one we call it as appendicular skeletal system. So axial skeletal system, if you are taking, it includes the skull, the sternum, ribs, these all parts, the pelvis, that part will be come under the axial skeleton. So the other limb structures, they contain the appendicular skeleton. So actually axial skeleton will be supported by the appendicular skeleton. So if you consider axial skeleton, around 80 bonds will come. The other one, it will come around 126 bonds will come. So that part also please remember. Now, while we're looking on to the very important structures here, the skull, if you are taking, the skull structure, you can see that the 8 cranial bonds and 14 facial bonds, right? And the skull bonds, if you include, it includes the 6 more ear ossicles, and below the buccal cavity region, there is a bond called hyoid bond, right? Now the sternum, you know very clearly that. Now look into the ribs. So there are 12 pairs of the ribs. Okay. So you know that generally speaking, ribs, if you consider, they are connected with the sternum. So how is their connection? Ribs dorsally connected with vertebral column, and ribs ventrally connected with our sternum. Here. So we have 12 pairs of the ribs. In that first 7 pairs, we call it as true ribs because they are obeying this particular rule. They are ventrally connected with the sternum and the dorsally connected with the vertebral column. So that's why we call them because as they are obeying the rules, we call it as true ribs. But the 8, 9, 10, if you take them, they are not completely obeying the rules. So we call it as false ribs. So why? What is the rule they are not obeying? Actually, if you look down, they are not directly attached with this particular sternum. Instead of that, they are attached with this particular seventh ribs only. So why we call them as false ribs? Because the 8, 9, 10 ribs, their ventral attachment, if you are taking, it's not into sternum. They are attaching just into the seventh ribs. Clear? So ventrally attached to seventh ribs. Clear? And now looking on to the 11th and 12th, they are freaky type. They are very freaky. They are not at all obeying these rules. So you call them as a freaky person, you call them as floating ribs. Here the false ribs at least making some attachment. This freaky ribs, they are not making any kind of attachment the ventrally. So you can say that these ribs are floating ribs. So the 11th and 12th ribs, if you are considered, dorsally they are attached with the vertebral column attachment is there, but ventrally with the sternum, they have no relationship. They are totally free. They have no attachment. So we call them as floating ribs. Clear? Now, after that, while we look into the next part, we can see that it is about the girdles. We have two types of girdles. So one we call it as pectoral girdles. Pectoral girdles, we have the two bonds, mainly the scapula and the clavicle. Scapula is dorsal to the thorax, and this clavicle we call it as collar bone, right? And the scapula, if you are taking, there is a cavity called acromion cavity. In this cavity only, who will attach? The head of the humerus will attach. So the head of the humerus is this only. This head of the humerus will attach to the acromion. So you can see the acromion also. This is acromion. So the head of the humerus will attach to this acromion. Here. So this is the scapula. This is the humerus. So this acromion of the scapula, they will be attached with the head of the humerus like this. Okay. So that part, please remember. So that's very clear. Now, similar to the pectoral girdle, you can also see the pelvic girdles. Here in the case of pelvic girdle also, you can see the three bonds here. It's also called as coxal bond. So what exactly we call it as? What are the three bonds present here? So look into this. Right. So you can see that this is the ilium. So they contain the three bonds. One is called ilium. Other one is called ischium. The other one we call it as pubis. So listen everyone. This is the ilium, ischium, and the pubis regions. Okay. So everybody listen here. This is the ilium, ischium, and the pubis. And you can see that there is a cavity present here. This cavity we call it as acetabulum. So what is this cavity in the pelvis region called? Acetabulum. So into this acetabulum only who will attach? Into this acetabulum who will attach? Head of the femur. Head of the femur will attach into this acetabulum. Clear? That part also, please remember. Right? So pectoral girdle and the pelvic girdle is also very clear. So we also talk about the axial skeleton. Now we can move on to the next part. That's our final part of this lecture. Right. The next thing we are going to see about the joints. We have the different types of joints. Joints means nothing but where the two bones are meeting each other. I had shown it with the scapula and this humerus, where two bones join each other. So listen. So you take one bone here. This is the another bone. So this is a joint, right? Now listen into the next part. We have actually three types of joints. The first joint we are discussing here, that's regarding the cranial bonds. You take the cranial bonds. We have the different types of bone. Okay. So you take the our frontal bone. This is the temporal bone. This is the parietal bone, the occipital bone. So just imagine, in the case of this cranial bonds, joints, we call it as fibrous joint. They are immovable joints. Now, while we're looking on to the next, that is the in between the vertebrae. The vertebrae you consider, you can see that this vertebrae you just consider here, you can see that this in between the vertebrae, we have a joint, and that joint we call it as cartilaginous joints. The fibrous joint in between the vertebrae, what you can see it, we call it as cartilaginous joints. And there is another joint, please remember, majority of our bonds are like that. There is another joint, and that joint we call it as synovial joint. Why we call it as synovial joint? Because it contains the fluid called synovial fluid. Okay, there are many examples for the synovial joint. The first are pivotal joint. Pivotal joint is what? It is in between the C1 and C2 vertebra. Gliding joint. Gliding joint is in between carpal bones. Now, the saddle joint. What is a saddle joint? It is in between carpal and thumb of metacarpal. Right. So then ball and socket joint. So pectoral girdle, pelvic girdle, these all you can consider for that. Right. Now there is hinge joint. Hinge joint, they will be allow only the one directional movement in the back and forth. So main example it is elbow joint, the knee joint, etc. Clear? Now moving on to the next one, please understand what we have to discuss, the very last part. So everybody listen. The very last part we have to discuss about the disorders of the muscular system. The first disorder, it is myasthenia gravis. So myasthenia gravis is a autoimmune disorder. Our immunity will act against us. Basically affecting neuromuscular junctions. So myasthenia gravis basically affecting neuromuscular junctions. So the next disorder, it is gout. Gout is happening by the uric acid deposition in joints. Why this is happening? This is happening because of disorder or the problem in purine metabolism. Purine metabolism problems. So remember that. So gout is uric acid deposit in the joints. Why this will happen? Because of the purine metabolism, some problems. Now, the next one is osteoporosis. In this, there is a demineralization of the bone. Demineralization of the bone. Okay. So, bone will get demineralized. That is what we call it as osteoporosis. While they get demineralized, listen, what will happen? They will be start to get some pores. So that's why we call it as osteoporosis. Females are more prone to get osteoporosis. Females are more prone to get osteoporosis after menopause. Clear? So they will get osteoporosis. They are very prone after the menopause because during the time of menopause, what will happen? Decrease estrogen. Decrease estrogen will decrease the calcium absorption. Clear? That's the reason the demineralization will happen and the pores will be created. Clear? So the next one, it is the arthritis. Arthritis simply means it is nothing but inflammation of the joints. But there is another arthritis. You should take it as important in the term of, in case of exam point, they can ask that. Rheumatic arthritis. Rheumatic arthritis is an autoimmune disorder only. Remember that. All right. Now moving on to the next one, the last one, that is tetany. Tetany will happen, decreased calcium in the blood. I would like to add the last and final point of this lecture. Tetany and the tetanus, do not be confused. Tetanus is the bacterial disorder getting by the Clostridium tetany. Tetany is the disorder, or it's a condition, the decreased calcium level in the blood. So both are different. Tetany versus tetanus. Tetanus is a bacterial disease causing by Clostridium tetany. You might be heard about locked jaw syndrome. The jaws cannot be moved, right? So both are very, very different. So that's all today. Almost every concepts we have covered in this chapter. So thank you very much. In the next zoology class will be about the excretion. That chapter will discuss in the next day. Right. Thank you very much, all of you. Bye.